I'm Neil Spooner, the chair of the Patient-Centric Sampling Interest Group, and I'd like to welcome you to today's online workshop, A New Era in Clinical Diagnostics, the Power of Self-Sampling. As I hope you're aware, this is a two-part workshop. So today we're going to be having some presentations. I'll talk to you a little bit about that, to you in a minute. We'll be then having a panel discussion covering what are the challenges to the more widespread adoption of patient-centric sampling for clinical diagnostics. And then in part two on April the 23rd, we will be going into solution mode and talking about how we address those challenges and what actions we should have going forward. For those of you registered, you will be, after this event, you'll be receiving an email with your link to the event on the April the 23rd. Just reminding you, if you're interested in the US-based events, you can register through the PCSIG website. They're scheduled for April the 30th and May the 7th on a time zone that's a little bit more compatible to our North American colleagues. So without any further ado, I'd just like to introduce you quickly to all of our presenters and panelists for today's workshops. So we'll start off with Vincent Molenaar. Vincent, are you there? Can you just quickly say hello?
Yes, of course. Hello, everyone. My name is Vincent Molenaar. I'm affiliated with Greiner Bio1, but also the Leiden University for my PhD in microsampling diagnostics.
Thank you, Vincent. John, could you quickly introduce yourself, please?
Hey, everyone. My name is John. And I'm a patient. I'm affiliated with the Patient Involvement Community United.
Thank you, John. Our third speaker today is Joseph Burt. Joseph, please could you introduce yourself?
Thanks, Neil. Hello, everybody. Thanks for joining. I'm Joseph Burt. I'm the head of diagnostics and general medical devices with the UK regulator, the MHRA.
Thank you, Joseph. And then our panelists. Sharon, could you introduce yourself, please?
Yes, I'm Sharon Geaghan. I'm the chair of the IFCC's microsampling work group, part of the Emerging Technologies Division, and from Stanford University School of Medicine in the US.
Thank you, Sharon, and thank you for getting up so early this morning. Next panelist, Tim Woolley.
Hi, my name is Tim Woolley. I'm the Chief Scientific Officer at Inuvi Diagnostics in the UK, and we've been doing capillary blood testing for about 7 or 8 years now.
Thank you, Tim. And our final panelist, Rachel Carling.
Hi, I'm Rachel Carling. I'm a consultant scientist at Synnovis, which is the pathology provider for Guy's, St Thomas's and King's College Hospital in London. And I'm also chair of the UK Metabolic Biochemistry Network.
Thank you, Rachel. Just before we get started, I just want to say thank you to all our partners and sponsors. Their sponsorship of PCSIG means that we can run this online workshop for free. We can run all our webinars for free and basically run our website and everything. So thank you to all those sponsors. Without whose support, we just simply wouldn't be able to do all the exciting things we're doing. So as I say, the theme of the event today is a new era in clinical diagnostics, the power of self-sampling. Today's first event is scheduled for an hour and a half, and you'll be having 3 presentations from Vincent, John, and Joseph. So Vincent will give a general background to patient-centric sampling. John will look at this from the patient's point of view, and Joseph will look at this from the regulator's point of view. We'll then be joined for a panel discussion after those presentations, where we will be working to identify the challenges to the routine adoption of patient-centric sampling for clinical diagnostics. And it's at this point that you, as the audience, get to join in. So please submit your questions, your comments, your suggestions through the chat function that we have here. And please note that this event is being recorded. It will be placed on the PCSIG YouTube channel probably next week. So it's there for you to review before our part 2 on April the 23rd. And we'll probably run, depending on how many challenges we come up with during the discussion, we'll probably do a panel vote on our LinkedIn group after the event to help prioritize what we talk about to do with solutions on April the 23rd. So with no further ado, I'd like to hand over to Vincent, who will give us our first presentation.
Hello everyone, my name is Vincent, and today I'll be presenting to you about patient-centric sampling, its concept, because this may be a new era in clinical diagnostics, and I will also cover its challenges that need to be overcome in order to make it a new standard in clinical practice. But before that, I would like to start with the reason why, because as you can see here on the slide, this is a very common procedure in healthcare, which is collecting venous blood by a healthcare professional, which is called venipuncture, of course. And while this has been an invention some time ago, societal demands and society has changed over time. And so the key question here also for this webinar is that, is venipuncture, did it keep pace with today's standards? And that's because this is an important element, because if we look at the analyzers that were used back in the day in the lab, they needed milliliters of blood for the analysis of the parameters. And so blood was collected in the evacuated tubes and then shipped to the lab, which you can see here also on the picture. So we needed a lot of blood back then, but today we need only microliters of blood due to modernization in the lab and lots of advancements, technological advancements. And yet, still, a lot of blood is collected from our patients, while actually not all of that blood is needed. Because if you look here from a paper, it's clear that labs collect much more blood than they actually need. And this volume, the blood volume that they collect on average, may even exceed 40 times the necessary amount, which is it's a lot, of course, to think about. And the majority of that sample is then discarded, because if you look here on the picture, this is the same tube from a patient before measurement and then after measuring 14 parameters on the Cobas instrument. And so it's very hard to tell if there's a difference between the two. Not from a picture, but it's also clear to see, of course, that the blood that is still left in the tube on the right-hand side will be discarded from this patient. And this not only applies to the single patient, of course, blood collection takes place a lot. And therefore, if you try to extrapolate that to North America and Europe, 25 million liters of blood are discarded on an annual basis, which is something we cannot close our eyes to, because it's blood that it should have been still in those patients. It's a very precious matrix, and if it's not needed for the analysis anymore, it should also not be collected, in my point of view. Of course, many healthy patients can there's little adverse effects that they experience from blood collection, but it needs to be mentioned that there are some adverse effects on especially fragile patients. For example, patients that stay 3 days or longer in the intensive care are at higher risk of developing adverse effects like anemia. And one of those reasons is the frequent collection of blood. And so, especially, of course, those fragile patients, they end up in the hospital, and blood is collected on a frequent basis over there. So this is not a good combination, especially for those patients. And also to say is that this is also definitely not a very patient-centric approach for those patients. If we look into the definition of patient-centric sampling, is also defined by the PiCC, and that is collecting high-quality biological samples. So it can be saliva, blood, urine, etc. anywhere and at any time. So it will be very decentralized. And usually it's also based on smaller blood volumes because we do not need this amount of blood anymore for the analysis as the technology continues to advance. Some of the advancements that are mentioned for this new technologies for blood collection are accessibility. And so this is not only in geographical reach for some people living remote, it's also with regards to equality for different patient groups. Think of needle phobic patients or people with a learning disability. It has been proven that they either postpone their blood work or not even do it at all, which is not accessible. It's not working in accessibility for them, of course. Then also because those newer technologies, they also provide a more convenient approach because they are more time and place independent, and therefore they're also less time consuming. And also proven less painful and distressing for patients, which could be an advantage also, especially for needle phobic patients. Now, if we look into the different technologies available within the patient-centric sampling market, there's actually two ways of collecting blood. It's either a liquid form or it's a dried form. And so liquid can be collected into a tube with a preservation fluid, without a preservation fluid, or with a preservation fluid. But it can also be collected only the serum or the plasma for stability purposes. For dried collection, you can also do dry blood spots. Heel prick is a very common example for pediatric. And there's also dried serum spot, where the blood is collected on a special paper, lateral flow paper, where the serum is separated from the red blood cells, which can sometimes be helpful for downstream analysis. Now, some examples, if we look into this chart, there are examples for liquid fingerstick collection, which some of you might be very familiar with, but this is an important one to mention. Here's, for example, preservation fluid in the blood collection tube. It's Labanovum, or the Gataco. It's a technology that can collect only the plasma. Also, I do want to mention here the automated venous blood collection, because I see this also as a more patient-centric approach, because it helps in improving accessibility with regards to placing those robots in certain more decentralized locations. Of course, it's not for everyone, but that's with any technology. And these instruments also can use lower volume evacuated tubes, which also helps with reducing the amount of blood being collected. Now, also, if we look into another liquid form of blood collection, which is more from the lateral upper arm, usually collecting around 1 milliliter of blood. These are devices that create a vacuum, and so they only collect a capillary sample. So there's no sensor inside, it's not a point of care testing, and it takes around 2 to 3 minutes for a capillary collection of the sample, and then the sample is shipped to the lab for analysis. On the right-hand side, you can also see examples of dried collection, which can be based also on the upper arm or can be based on fingerstick. This is an interesting one also to mention. It's also the dried serum spot. So you can see over here that the serum is separated from the red blood cells. So these devices that I just showed you are actually all examples of self-sampling. And sometimes there's a little bit of confusion between self-sampling and self-testing. Self-sampling is collecting the sample and then shipping it to a lab for analysis. And self-testing is collecting it and testing it immediately. And so this is a key difference that can be made for the different technologies. And I just showed you self-sampling technologies because we see a lot of advancements also over there. If we put this into perspective, you can see that the patient receives the self-sampling device at home or can be used in a clinic, and then samples the blood from, this case, the upper arm, and sends the sample to the lab where the analysis is done and the results are received. And what this brings is that it creates a situation where the blood draw is time and place independent. There's also no need for trained phlebotomists or draw locations if it's at home. And for the patient, he can do it without the waiting room, without extended periods of fasting when needed, or taking time off work. Some key considerations that I do want to highlight, as our webinar is also about challenges here. Of course, for the patient, this technology needs to be safe and very easy to use. It needs to be usable to them. Otherwise, it will also not end up with high quality samples where, of course, the analytical performance is important. At the same time, it also needs to be reliable. If there's a high failure rate for collecting capillary samples by a patient, him or herself, that is also not ideal because it's repeated sampling needed. And also on the right-hand side, of course, it needs to be compliant with the regulatory standards that we know. And its logistics also need to be very of high quality to make sure that the sample is not influenced during the transport. So if we look into this a little bit, we zoom into this a little bit, I see that there is a couple key messages for this new patient-centric sampling, is that it's more than just a device. You can see it more as a whole system, because if you look here at this this picture on the top left, it's a whole system, and that's also the way we should look at it, because if the patient collects a sample, it also still needs to be shipped to the lab. So the time to analysis is relevant. Also, of course, the transport systems that need to monitor temperature, humidity, or shocks. These are all very important to make sure that labs communicate high quality results, because that's, of course, the whole purpose of diagnostics, to gain insights from those results, and those should therefore be accurate. And the reliability rate, of course, and the handling of the samples in the lab also needs to, it's a different approach. And so there's a lot of new pre-analytical variables. And so all these different building blocks should work together to create a good quality system. And as mentioned, of course, the analytical quality is a prerequisite in order to communicate accurate results, and therefore the stability of the sample is important. And ways to do that are centrifugation, but of course there's knowledge gaps around delayed centrifugation or maybe in-field centrifugation directly at the patient, but this is maybe more technologically challenging. And then, of course, the hemolysis degree. These are all important factors. One point that I also want to highlight is that not every patient is the same. So there could also be differences between those patients and variability. So this is all important to mention, not to forget, of course, the regulatory bodies that this need to be compliant with. And this applies to the devices, but it also applies to the instrumentation companies that need to clear capillary claims, for example. And then lastly, I also want to mention that patient instructions are are essential. It's very important to make sure that the patient feels confident enough to collect his or her own blood. So therefore instructions are very important to prevent underfilling or overfilling, or maybe interferences such as lotion or sunscreen. And well, there can also be device specific challenges here, because sometimes the device can only collect one tube. In general and in summary, I think these are all very important challenges and definitely we should be talking about it. And they will eventually be overcome, because I really believe that the future of blood sampling will be small. Thank you very much.
Thank you very much, Vincent. Really good introduction to the area of patient-centric sampling. We're going to do, just for those in the audience, a reminder, please hold your questions and comments to the panel discussion at the end, where we're really going to be looking at what the challenges are to the routine adoption of patient-centric sampling for clinical diagnostics. But before we go on to that, we've got a couple more presentations. The next presentation is John from PiCC United, and take it away, John.
Hello, everyone, and thank you for joining. My name is John Fabule, and I represent the Patient Involvement and Collaboration Community United as a patient advocate. Today, I will be speaking from a patient's perspective about the need to involve the patient voice more in clinical diagnostics, and specifically the work done by the lab. The broader laboratory community being our main audience for today, while also recognizing that many others who support the diagnostic pathway are on the call. Before I go any further, I want to start with one striking observation. When I looked for data on how many different types of laboratories work with patient partners on a permanent, ongoing basis, not just for a one-off project or occasional feedback, but as a routine part of how tests and devices are developed and improved, I found surprisingly very little published evidence. And that in itself tells us something important. It tells us there is a gap. And that's across laboratories, we still do not have good evidence that this is routine practice. For example, one useful 2024 evaluation found that even in a research institute with an established involvement program, only 43 out of 83 laboratories, about 52%, had any patient or public involvement. A 2021 scoping review reached a similar conclusion, finding only 32 published reports of patient engagement in preclinical laboratory research over a 50-year period. And finally, a 2025 interview study, again, described engagement in this space as limited, with researchers pointing to the need for a broader cultural shift before it becomes normal practice. So the message I want to open with is this. We still cannot confidently say this is standard practice. Where it happens, it is still not consistent, standardized, or widely embedded. And for me, that is not criticism. It is an opportunity. Bringing the patient voice into the lab consistently and on an ongoing basis is still not common enough, but it is necessary. It is necessary because patient voice helps us understand where good science can still fail in real life. It tells us where instructions are confusing, for example. It tells us where sampling becomes difficult or may become difficult. It tells us where packaging, timing, transport, or even dexterity problems interfere with success. It shows where a technically strong test may still break down before the sample ever reaches the analyzer. And that is why one-off engagement is often not enough. When patient partnership is systematic, ongoing, and built into the development pathway, the patient voice becomes more useful, more scientific, and more actionable in my view. It becomes something you can track over time and compare against, for example, reject rates, redraw rates, usability issues, shipping failures, and borderline result interpretation. It becomes something that strengthens your quality system rather than sitting outside of it. Patients do not experience diagnostics as method validation protocol, a stability study, or an analytical performance study table. We experience diagnostic as uncertainty, waiting, confusion, delay, access or lack of access, as the case may be, and of course, sometimes as reassurance or life-saving action. And that is why I want to make this personal. And I would like to begin with a personal story, because for me, this topic is not theoretical at all. Some time ago, I developed what I assumed was an indigestion. It felt like chest discomfort, but it was not resolving over several days. It was nothing dramatic, but it did not feel right, and it was not going away. Eventually, I went to a local walk-in clinic, partly for reassurance. They did an ECG, and they checked my cardiac enzymes, specifically troponin. The result was not off the charts. It was in the high normal range for the troponin, borderline enough that without the right clinical context and without confidence in the test itself, it could easily be dismissed. But because the test had been designed, optimized, and validated to be reliable, and because clinicians could trust what that borderline value might mean, I was escalated for further evaluation. Other workup showed that I had severe coronary disease, and I was scheduled for open heart surgery, ACABG, cabbage. While I was waiting for surgery, I went into cardiac arrest, and it took about 35 minutes to bring me back. A less than 1% survival rate for that type of cardiac arrest, a pulseless electrical activity after 30 minutes. But I survived. And I can tell you from lived experience that the reliability and interpretation of that lab result helps set a chain of decision in motion. And that chain definitely changed my outcome. But why could the healthcare professionals trust it? And it's definitely because the lab community had done the hard work. They had optimized assay sensitivity, they had controlled imprecision, they had validated cutoff values, they had shown that a rise or fall on repeat testing actually means more than just a single static result. That is why a borderline troponin result often triggers escalation, repeat sampling, ECG review, clinical correlation, and sometimes urgent cardiovascular evaluation, rather than simple reassurance and discharge. So, when I look at lab teams, I do not see people who are far away from patients at all. I see people who are quietly and often invisibly help decide whether patients like me get a second chance. And that is why the work the lab does matters, and why it matters so much to me. The way I picture and explain the connection between patient sampling experience, patient voice, and work done in the lab is this. I'm reminded of patients boarding a plane when they collect a sample at home. They are busy. They may be anxious. They may already not be feeling well. The pre-analytic variation is the weather. That variation might be delays, heat exposure, underfilling of samples, mix-ups, or even hemolysis. The method and quality control are the control tower. Those responsible decide what is safe to land. In other words, what is safe to report and what needs to be rejected and redone. The clinician is the pilot making the final decision. And of course, the patient voice is the weather report. It tells us what conditions are actually happening in real life. And that is what makes it so valuable. So why does the patient voice matter? Here is a key point. Even a perfect essay can fail in real life because of what happens before the sample ever reaches the instrument. Patient voice matters because it helps us understand and reduce real-world failure points. This might be underfilled samples, mislabeled specimens, delayed shipping, temperature, exposure, contamination, poor mixing, incorrect timing, confusing instructions. And these are just some examples. Or very human barriers such as pain, embarrassment, low vision, language barriers, or even limited dexterity. When those things happen, they do not stay invisible at all. They show up in the lab as more rejected samples, more redraws, more inconclusive results, and more noise in the data generally. So, for me, patient insight is not about relaxing standards at all. It is about making it more likely that real-world samples actually meet the standards you already require. So the patient voice does not weaken quality. For me, it strengthens quality. And I know when I say the lab, I know that means many different things. Because some labs are trying to discover new science, some are building or validating new tests, some are supporting clinical trials, some are running hospital testing every day, and some are helping patients use testing closer to home. So I'm not saying every one of those labs needs the exact same patient role. What I'm saying is that whenever the patient experience affects whether a test is used properly, whether it is trusted, understood, or even completed at all, the patient voice matters. Across the field, we still do not have strong evidence that ongoing, built-in, permanent patient partnership is standard practice. And that is why this matters. Because if patient voice is only appearing at a few touch points, lab may still miss the real-world problems that decide whether a good test or device succeeds or fails. Common question that often come up. First one isn't patient voice subjective? How can we use it scientifically? And my answer is this. Treat it like any other input. Collect it systematically, look for patterns, and convert those patterns into testable conditions and measurable metrics. For example, such as reject rates, hemolysis rates, insufficient sample, or delayed shipping. For example, if 7 out of 10 patients say, I can't tell when I've collected enough, to me, that is not just an anecdote. That is a design signal. And in the lab, that signal may show up as insufficient volume rejections. So what do you do with that? The design could be addressed. The level could be made more obvious. That insight could be converted into validation work. Low volume samples could be tested, for example. Reject thresholds could be defined. instructions could be updated, and then the effect on insufficient volume rejection rates could be measured. And that, to me, is how subjective experience from patients can become scientific improvement. Another question we get is, how do we stop this becoming a never-ending wish list? And I think that's a fair question. And my response to that is to set boundaries. You can say, for example, we are focusing on changes that improve reliability, reduce burden, and expand access without compromising analytical vigor. Our patients usually respect those kind of boundaries when they are explained clearly. So, for example, a patient may say, I want results immediately, or I wish these involve fewer needles. Now, the exact request may not always be possible. Some limits are set by the biology of the test and by what is needed to produce a reliable result. But the value of that comment is that it shows you where the burden sits for the patient's point of view. So it may not mean you can remove every step, but it may mean you can make the process less confusing, less stressful, and easier to complete correctly through perhaps clearer instructions, better timing guidance, simpler kit design, or more intuitive packaging. And these are just examples. And in summary, you could just say, we listen broadly, but we act narrowly on the highest risk and highest frequency failure points. The third common question we get is, for those who are focused on assay validation or method development, how can patient insight realistically change validation plans? And to that, I say, patient voice tells us what real life looks like, so we can validate the method under the condition where small signals must still be trustworthy. So going back to my own story, cardiac troponin made a difference to my outcome, even when the result was in the high normal range. And that tells us that borderline results matter, especially when the assay has been optimized and validated at that level. So what can someone in assay validation or method development do? Add pre-analytic stress tests based on what patients say actually happens. So for example, room temperature versus a warm car, mild hemolysis or lipemia threshold, repeatability at low concentration near clinical decision points. These are just few examples. And we do get other questions on how many scenarios to test for and patient influence on interference panel, but I won't go into details about those due to time. Let me move quickly to something more practical, which is what does patient voice actually look like in the lab on a day-to-day basis. Patient engagement, for me, does not have to be large or complicated. A few focused actions can already make a real difference. And that may be where you start your focus. So you could, for example, organize a small patient partner group, do a sample journey review and ask patients to walk you through collection on their worst day, not their best day. Observe a usability run through before you lock the method. Add patients' reality into validation by including delays, temperature swings, underfills, or shaky hands, and then quantify the impact. Co-create instructions in plain language while preserving the scientific requirement. And one that often gets left out, which I would definitely encourage, is to include collaborations with patients in publications. And if you think time is short, I think the two places I would recommend starting are sample journey review and patient reality and validation. Those alone can go a very long way. Another common question is this: we do not have access to patients. How do we start? My answer to that is, start with what you already have. Look at complaint logs, for example, if you have them, or redraw reasons, call center questions, and even clinician feedback. That already tells you quite a lot. And then, of course, you can then move on to partnering with an existing patient group or network where governance is already in place. For example, patient communities like PiCC United or patients like me, I think the overarching principle, which is basic, is this: engage ethically, transparently, and with a clear scope in mind. Patient voice works best when it is structured, respectful, and used to drive measurable improvement. So I'll close with this. Patients do not experience diagnostics as a technical process. We experience it as worry, hope, delay, clarity or lack of clarity, as the case may be, and sometimes as reassurance or even life-saving action. To me, labs teams are central to that story. When you optimize an assay, define stability windows, reduce pre-analytic errors, clarify instructions, improve acceptance criteria, or refine threshold, you are not just supporting patient care, you are enabling it. So my invitation today is simple. Build patient reality into your quality system in a systematic way, not as a campaign, but as a routine, measurable partnership. Thank you very much for your attention.
Thank you so much there, John. It was very interesting and important, and some very practical tips there about how to bring the patient voice into your research and your clinical chemistry labs. So the final presentation before we move on to the panel discussion is Joseph Burt from the MHRA. Hi, Joseph.
Hi.
Hand it all over to you.
Thank you very much, Neil. Hello, everybody. I'm Joseph Burt. I'm the head of diagnostics and general medical devices with the UK regulator, the MHRA. I'd like to just maybe speak a little bit more from a regulator's point of view around the patient-centric sampling approaches from the device's point of view. So I'd like to start off with saying that sampling devices which are used for human specimen collections and in vitro examinations are regulated products here in Europe. So it's very important that we understand this basic understanding because it is described in the regulations itself. So here I've extracted the EU IVDR definition of an in vitro diagnostic device. And it clearly says in the red parts of the regulation of what an IVD device is a kit, an apparatus, and equipment that's going to be used for in vitro examination of specimens and for the following points that you can see on the screen. The important thing here is specimen receptacles are also considered to be in vitro diagnostic devices. And similarly, in the UK regulations, the similar words are there, but it is a separate set of regulations that applies in Great Britain. But you can see that the kit, apparatus, or equipment, which are going to be used for in vitro examination of specimens for the following purposes, are considered to be regulated products. This includes specimen receptacles. So hopefully you can already understand from this point of view that these sampling devices, which we have talked about and which are very important for this particular webinar, are regulated by the regulations in Europe. In the UK MDR, in the EU and Northern Ireland, it's EU IVDR. So what I'd like to just put forward is to describe the 5 principles that we would examine for making sure that patient-centric sampling devices are in place. So first and foremost has got to be no harm during sample collection. The procedure for collecting the samples itself, it could be a venous draw or other sample collection methods, must have the ability to not harm the individuals using it or collecting the samples itself. And that any particular risks associated with the use of the device must be reduced as much as possible, and should also consider the validation of the demographics of the users. So this is when I say the demographics being age or the population in which you're going to be deploying these devices onto. For example, pediatric cases shouldn't have needles in them, for example, but need to have other methods and collections. Similarly, for the other principle around specimen quality and quantity, Vincent talked about the huge volumes we go put into waste streams these days. 25 million liters of blood is a huge amount of blood that we put into waste streams. But importantly, the specimen itself needs to be of the highest quality that is going to be available for future and further processing of that sample itself. There needs to also have the sufficient quantity so that the quantities can be used for the processing of that specimen, but also potentially for other purposes, for example, retained samples. Importantly, the stability of the specimen must also be verified and stable, with evidence generated to demonstrate its stability. For example, we cannot require the specimens to be collected and be then stored for a long period of time without some kind of refrigeration. So the stability of the actual specimen needs to be considered for when you are taking that specimen and putting it into the sample receptacle. The fourth important principle is around diagnostic clinical performance. So as we have already seen from the regulations and definitions, these are for in vitro examinations. And so if you are collecting the samples itself and making sure that you are using those samples for in vitro examination, the performance of the devices, the IVD assays of the test itself, must be characterized with the sample collection approach that has been taken ahead of the processing of the actual sample itself using the IVD assay. The clinical performance, typically seen as the sensitivity or specificity, but other measures also apply, needs to be understood based on the sample collection device being used up front. As we have seen for all devices, especially across Europe, the post-market surveillance requirements also persist. So as your device is a regulated device, post-market surveillance requirements and vigilance will need to continue. Harm associated with the use of the device must be reported to competent authorities and regulators and ministries of health in several jurisdictions. Most importantly, the manufacturer of those devices must ensure that they are actively monitoring these devices in post-market controls. So let me just now talk about some of the challenges associated with making sure that these devices are actually put into practice. Fundamentally, the use of the device by the self-sampler or the user must be understood very clearly with simplified, easy to understand instructions. Training may need to be given, be it, for example, in an online platform where a video could demonstrate how something could be taken as a sample, or through proctoring of that particular sample collection method. As I've mentioned already, some of the importance around stability, but also stability of that sample and the device itself during transportation. And this is where packaging needs to be really understood and tested and verified. Similarly, other regulations apply for the transportation of these biological samples following the UN 3373 principles. As well as those transportation principles, there needs to be a consideration of the specimen being received and logged once received into the laboratory. Mix up or control of the sample during further processing must be considered. Similarly, waste streams, for example, the 25 million liters of blood that's being put into waste streams, must be further considered for the impacts of those wastes. Hazardous and infectious waste disposal must be considered upfront. And most importantly, is around patient consent. The patient must be consented to be able to give the sample, but also to be able to use the device for self-sampling. Let me now talk a little bit more about some of the requirements specifically for validation and quality assurance. So validation of sampling devices is mandated in many jurisdictions, including in the EU IVDR, but also in the UK medical device regulations. Robust validation, that is examination of the sampling device, must ensure consistency around the accuracy and reliability of the collected data during all the self-sampling procedures. Similarly, the validation and validity and reproducibility of the results during the examination of the sample must be fully characterized. So regulators often look at how well the performance of these devices, the IVD device, is going, and it's really dependent sometimes on the sample collection method and devices up front. So there needs to be a correlation between demonstrating validity of the sample collection device, the IVD assay or test, and the production of the results that is able to be validated and reproducible. Fundamentally, quality systems are important when considering sample collection. There are certain standards that need to be followed, and many regulators and certainly competent authorities and bodies that do conformity assessment will look at the quality systems that have been instituted within the development and the manufacture of the self-sampling device. We need to make sure there's consistent high-quality outcomes that we are always looking for with the self-sampling device through the quality management system and the use of standards. So importantly, I'd like to just recap on 5 very important elements around from what a regulator would look at for self-sampling devices. It's around being the device is regulated. So medical devices are regulated products, and a self-sampling device is a regulated medical device. Number 2, there must be no harm to the users or the patients using the sample device itself. The challenges must be overcome in the practical and technical challenges that I've spoken about already, and the consideration of post-market surveillance and vigilance activities to ensure that there is ongoing monitoring of the device use by the manufacturer and the reportability of any harms associated with the use of the self-sampling device. As I already just mentioned, it's really important that the quality and the standards of the self-sampling devices are fully adopted into quality management systems and demonstrated through assessments. Finally, the important aspect in all of the discussions here today is around the use of the self-sampling device in a validated manner, consistent with the use of that self-sampling device in further downstream processing. For example, in the use of the sample itself in vitro examination of the specimen for disease or conditions. And importantly, these, as we already have seen, are intrinsically within the use of the decision-making for the clinician for patient management purposes. So we need to have the confidence that the validation and the evidence of these self-sampling devices are appropriate for the intended purpose, but also making sure that they do not cause harm, but also ensure that the reliability and quality of these products do not hamper the decisions that the clinician will make as an outcome of the use of the IVD. And so with that, I'd like to pass back to Neil, and thank you for listening.
Thank you, Joseph, and thank you to all our presenters. So if we can all come back on screen, so and we can start the panel discussion part of this workshop. So hopefully that's given everyone a bit of a background on what patient-centric sampling is, if you were not aware, how we might best engage the patient, and what we might need to think about on the regulatory front. What I want to go into now with the panel and the people joining us online is talking through why we aren't doing this routinely already. So there's obviously something that's stopping it. You know, a bunch of these technologies have now been around for quite some time, and some of them are new, but some of them are not. So there's obviously something stopping us using these self-sampling technologies for routine clinical diagnostics. So what are those barriers? What I don't want us to be moving into in this part of the discussion is how we solve those problems. We're going to prioritize the challenges, and then in 2 weeks' time, well, a week's time, sorry, on April 23rd, we're going to be talking through how we solve those challenges. And just a reminder to everyone that's attended this meeting, you'll be automatically, after this event, receiving an email with an invitation with your link to attend the event on April the 23rd. And we'll be sharing the output from this webinar on our YouTube channel, hopefully next week. So does anyone want to jump in as to what their thoughts are as to what the challenges are to more routine adoption of patient-centric sampling for clinical diagnostics?
I can go first if you like. So my lab, we've been doing capillary blood testing for about 7 years. We were one of the first labs in the UK to get accreditation, ISO 1.19 accreditation, and we do probably something around the realms of about 1,500 capillary samples a day, maybe 2,000 capillary samples a day. But it's extremely manual. That's a major problem in that there's a lot of pre and post analytical platforms aren't designed to handle capillary blood samples from the primary tube. So you often have to aliquot, which obviously is again a problem because you've got a small amount of sample. So it's a very manual process, or at the moment it is a very manual process. The other issues are around validation. So you have to validate, not verify. And how you do that, you can use different publications like EP35, for instance, which we use as our go-to document. But it's not the data is not in the IFUs. Now Roche have recently changed that and added a few tests to be CE marked for capillary blood, but that's obviously another issue. And then ongoing for that is experience in using capillary blood in a major lab. So if it's a small number, it's quite easy. When you're looking at thousands, it becomes a bit more of a process.
Neil, you're on mute.
I'm trying to take notes and therefore not click in the background. Thank you, Tim. So anyone got anything to add to that? You know, one of the main barriers being the very manual process in the lab and
Right.
the lack of link to automation.
To add on to Tim's description, I having been for about 10 years, I did nothing but direct a pediatric lab. And of course, by definition, you have a high proportion of heel sticks. We're serving the highest level of neonatal ICUs. And, you know, just to add on in addition to the increased manual labor of aliquoting and the problems with quantity not sufficient and so forth, there's a very real patient care risk of mixing up samples because there is so much, manipulation and so forth. And on a large scale, that really adds a lot of risk if it's not managed well.
Yeah.
Okay, thank you.
Could I chip in, please, Neil?
Yeah, absolutely.
Yeah, so I think in my experience, two other things that are quite difficult to resolve sometimes are the logistics of getting the devices to the patients in the first place, receiving them back in the laboratory, and then how those samples are reported, so the IT issue, which isn't just limited to microsampling, to be fair, but they were two of the things that we struggled with.
Okay, thank you, Rachel. So that's logistics, getting devices to patients, getting samples back to the lab, and data
Booking,
out.
yeah, and also booking in, and then how do you know that the sample's been sent? It's not like it's not requested in the same way, is it? So you don't know that samples have gone missing until it's too late. It's all sort of tracking and logistics pre-analytically.
Thank you.
If I may add here also is that, as I mentioned, I see this as a new pre-analytical process also. And because currently, of course, a lot of diagnostics are based, the main matrix is venous blood. And this is also it's a golden standard, you know, exactly how that takes into place. But with the newer self-sampling approaches, of course, there's also a need for a new pre-analytics platform where you instruct your patient, but also that a lab, as Rachel mentions, knows that a sample is coming, because they are also compliant to know and to track and trace their samples. But also I see another challenge in this is that if something goes wrong, because we're not working with healthcare professionals, we're working with patients. So these are lay people. If something goes wrong, who is then responsible? So I see this as a common issue that's raised. Who's responsible if something goes wrong? So that's also something I would like to add.
Thank you, Vincent. I think that's a really good point. You know, if something does go wrong, who does the patient contact to know what to do there?
Yeah, and from a patient perspective, I think one of the things that I would like to point out is the usability barrier, because a simple device does not mean a simple experience for a patient. So when patients struggle, to use the device appropriately, or they're struggling because of limited dexterity or poor vision, or they're elderly, I think it's a problem at the end of the day. So if that can be addressed, I think it will go a long way to patient trusting and actually using it more, and that will actually improve the adoption as well.
And then one of the other things is the sample matrix, isn't it? And whether the microsampling result is going to agree with the laboratory version of the result. And obviously, something like creatinine is a good example of that.
Will you expand a little bit on that, Rachel?
So if you have a lab that obviously is churning out serum creatinine routinely and giving the EGFR along with it, then you suddenly start doing, say, tacro and creatinine by some type of remote sampling device. The creatinines are going to be different, so they either need to be reported using a separate test code, which is probably the simple way to do it, or as we ended up trying to do it in the middle of COVID, you end up reporting a serum corrected result, which is not what you would necessarily choose to do, and creates a whole host of separate problems. And also in terms of the sort of the activation energy to get the process set up in the first place, that often requires looking at different reference ranges, or could require looking at different reference ranges for the new sample type, and doing more extensive validation work to make sure that you understand how it differs to the lab-based, or I guess the sort of the traditional way of measuring that marker.
Thank you.
Maybe to add on the specifics of the analytes, is that also what I see is that, well, if you look into the literature, and a lot of progress has been made already, is that, we're talking venous, method comparisons, venous, capillary. For many analytes, actually, they're quite comparable, but of course, the blood in the human body has a function to, exchange certain metabolites and everything. So there are definitely analytes that need a closer look for comparability of the analytes. But also, I think what's important is that the method of collection is different. So as I showed, there's the finger stick examples, but also there's a more standardized approach from the upper arm. And so all these different approaches to microsampling, I'm now talking about liquid, they also have a different effect on, for example, hemolysis degree. So it's been shown that there's a different hemolysis degree level for the different capillary blood collection methods. And so I think this is also maybe a little bit uncomfortable for labs to work with that, because there, of course, is not one particular golden standard yet for capillary sampling.
Yeah, I mean, from an operational standpoint, for obviously what we do is that sample is going to be in the post. We get everything delivered by the Royal Mail. You're looking at, 24 to 72 hours on standard. So whole blood stability evidence is what we've done. We've done lots of it, but there's more papers coming out supporting going out to, in some cases, 4 or 5 days. So at different temperatures, but that data is still, lacking. We're talking studies of 50, 60, 100, 150. So whole blood stability at different temperature ranges it will be ideal. And I think the other thing is that because the technology is quite new, we still don't know some of the nuances around contamination. Now, Vincent, you brought up a couple things like sun cream, hand creams, hormone creams, things that get onto the skin and get into the blood tube. So we've had years and decades of knowledge of venous blood and what that entails, but we haven't had as much detail and information on capillary blood collection and what patients can do. And at least with a venous sample, you've got a professional taking that sample, so therefore the education, the training's there. But with a patient, you've not got that. So the pre-analytical, the patient information, the IFU is critical, and it obviously has to be tailored to the client cohort rather than just a generic one, like a mixture of online, maybe in-person training at the GP for a first couple of samples, or and then insert as well is the way to go. But that balance has to change depending on the patient cohort.
Yeah, I'm just going to go back. So we've been getting quite a lot of comments from the online audience. So I'm just going to go and have a quick look through to see whether we're missing anything here. So concerns about reliable sample delivery. I think we've kind of covered that a little bit, but we need to make sure we capture that. So thank you, Julie. Let me just...
Maybe if there's something I may add here, Neil, is that here in the Netherlands, for example, we have something called Medical Post. And this is actually quite helpful, because what they do is they make sure that, this is specifically for biological samples. And so they make sure that the sample stays around ambient temperature and is delivered within 24 hours. And so this is very helpful for these microsampling approaches. And I think this is also maybe an issue in other areas of the world where, when we address the stability topic, this is something to think about.
Thank you, Vincent. And I think that's maybe something to talk about next week in terms of solutions, in terms of spreading the learnings of what's done well in one part of the world to maybe what other parts of the world could start to do. So I've made a note of that to maybe bring that up for next week as well. Thank you, Vincent.
On the challenges side, I have a question for Joseph. In the US, the FDA has a mandatory requirement for manufacturers in terms of post-market surveillance, but on the patient side, it's voluntary. And this is problematic in terms of actually having a lot of that information lost, because it's a voluntary and somewhat time-consuming reporting. And with more testing moving into the home and moving to the patient and in the patient's hand, that's a real challenge, I think. And I was inspired to write a piece about case reports, because case reports have fallen out of favor. A lot of journals don't even have a section to publish case reports, but we were finding a lot of interferences, and important problems were being sent to us as journal editors, that would otherwise be lost. And I was wondering in the UK if this aspect of post-market surveillance and the patient taking a greater role in that perhaps is something that is a challenge there as well.
It is an absolute challenge there, Sharon, and thanks for the question. It's a challenge that we've seen persist for many years. And in the UK specifically, we introduced new regulations last June that are now enforced to require manufacturers of self-sampling devices to constantly monitor their product whilst being in use and on the market. And it's the same in the EU. There's post-market surveillance regulations in the EU IVDR have been there since 2017. So it is now a mandatory requirement by law to monitor the use of the self-sampling device whilst your device is actually being used in the market by users.
So thank you for that, Joseph and Sharon. We have a comment here, and I think it's an interesting one from Frederick. His statement is, we find laboratories to be a bottleneck. What laboratories have validated large panels of standard clinical chemistry, such as... So I think what he's getting at is finding out what labs have what tests on what devices is a challenge. Does anyone have any comments on that?
In the UK, obviously we're UCAS accredited, so if you wanted to look up capillary blood testing, you can go onto the UCAS website, look under accredited labs, and then look up certain tests, and then it should show you on their schedule if it's accredited in capillary blood. And in the UK, I think there's 4 laboratories that are currently accredited for capillary blood. And that's a mixture of private and NHS. And
does that, Tim, does that then list what analytes are available, or does it just say you're accredited?
No, so in the schedule, on an ISO 15189 schedule, it has to tell you what test, the methodology they've used. For instance, we don't use Jaffa creatinine because we know that the creatinine method, Jaffa method, it's inaccurate for postal samples, so we use the enzymatic method. So it'll have their enzymatic method, the actual manufacturer of it as well, and if they're accredited for venous, whether it's serum, plasma, or capillary blood as well. So there's a couple of labs in the UK that have got plasma and serum. We're one of them for capillary as well as venous.
Thank you, Tim. Just looking at the comments from the audience. Amy, thank you for your comment here. How should labs approach the issue that not all testing can be collected at once? CBC and chemistry tests currently need different tube types, but most self-sampling devices only do one tube. So any thoughts about that whole thing, about needing different tube types? I'm not going into solution mode, but is that a challenge that people are seeing?
Rachel, you were saying something, but you're on mute.
Yeah, sorry. I was saying I think it's a really important point because we've had quite a few requests from clinicians saying, could you set up microsampling for, say, let's go back to TACRO, which is a familiar request that everybody's aware of. And we said, yes, we think we can. And then suddenly it was, can you do a creatinine as well, please? And then suddenly they wanted a full blood count and a potassium. And then suddenly you're in a very different, certainly a lab like mine, which is specialist testing, you're in a very different place. And often I find that's where people sort of drop the idea because it suddenly feels too difficult and isn't actually going to achieve what they want. Because obviously if you only get 1 or 2 tests, but actually you want the patient to have 3 or 4, then home sampling is not necessarily going to achieve what you're setting out to do.
Yeah, I think it sounds like a good change. Sorry, Tim, you were saying?
Yeah, so we've got a couple of people that use different devices that get 2 tubes. Not for FBC. FBCs are still currently probably the gold standard. If you can get a device that gets a reliable FBC, then you're probably going to take the market by storm. So we see 20, 30, sometimes 40% failure rates due to clotting on capillary blood. Although we've verified it. If you get a sample, it's a good sample. It's very comparable, and there's lots of papers on that. We can get, depending on the platform, we can get 20, 30 analytes, chemistry analytes, immunochemistry analytes from a capillary sample. We do get a client that uses lithium heparin for HbA1c and then on for chemistry tests. So there are ways and means around it, but a lot of that is dependent on your platform, because some platforms use a lot more for immunochemistry, for instance, than they do than others. And I think most of the chemistry analyzers use very small amounts, and that's kind of standard across the different ones. So there are the 2 tube thing is an issue. It can be overcome, but it's not for everything. I mean, very, very rarely will you get a fantastic, set of full blood counts from a capillary device.
I do wonder if that's actually one of the big barriers that we've got in terms of implementing microsampling, in the sense that what we're trying to do is use the routine clinical labs that are already set up to start running these tests. So we've all got lots of issues and lots of practical problems because we aren't designed to take these sample types, whereas Tim's lab is obviously doing really well at this. And I don't know much about your lab, so I don't want to make any assumptions, but it sounds like you are much better set up. So I wonder if actually what we need to do is change the question and not say, how do we get a routine chemistry lab to start doing all this testing, but say, what sort of lab do you need if you want to do microsampling? I don't know if that...
We've been doing it for a long time. We did a lot of during COVID. We did tens and tens of thousands. So we learn from that to do large numbers. But it's very difficult that we've got a lot of staff that do it. It's still quite manual, and the analytics are just coming in. We've just got a post-analytic platform now that can handle capillary samples. Yeah. We don't have a pre-analytic yet. We do know of a manufacturer that's bringing one out next year. So we'll look at it then. But at the moment, a lot of that is very manual. But we have a lot of staff to do that. I mean, I probably have 10 or 15 staff doing the numbers that in a big NHS lab, you've probably got 2 people doing.
Yeah. Yeah. So I do think that's quite an important point.
I'm just looking through the comments here. It's quite interesting. The attendees are talking to each other and sharing what assays they have and things. So it's quite
That's great. Could I ask John a question?
Yes.
John, your presentation was really incredible. I saw the initial reference that you put out showing the relative lack of engagement of patients in today's laboratory. And I wondered, again, with more testing moving to the home, with more testing in the patient's hands, it seems an imperative that this change. And I just wondered, in the time that you've been speaking and telling your very compelling story and your analysis, do you see any traction? Do you see any evidence that more patients are getting involved, or that folks on the laboratory side, who we traditionally have been solving laboratory problems with ourselves and not with the end user to a large degree, is that changing at all? Or how? We're not supposed to talk about solutions quite yet, but do you see anything changing?
Yes, I mean, it's a great question. I think for me, patient advocates like myself need to get engaged more in forums like this, and vice versa as well, because I think there's still that gap that people that understand what the lab is doing and know what patients are going through are actually going to speak to them, and then the lab as well going to speak to communities like mine, like patient involvement and collaborative committee. So I think when we have more of that cross-fertilization of ideas, it will even improve more. But right now, I think it's still at the early stages. I would say there's a little bit of movement, but again, because patients think they need to have that expertise to be able to speak confidently and credibly to the lab community, because scientific is not, and so you need somebody that really understands a little bit. I'm not an expert, what lab does, but at least I can understand when they speak. I've been a physician myself, so right. When we get more people who actually understand it too and can bridge that gap, I think we even get more adoption and more uptake. So to answer your question, I think yes, there's a small uptake. People are getting more interested, but I think we're not, nowhere near where we need to be in terms of folks like me actually going to speak more to an audience like this. And I must confess that even before my own health scare, I wasn't doing that. So it took something very, very dramatic to actually get me sitting off the sideline myself, to actually get more involved. And I think the more people like me speak to our community and a community like this, and we'll get more people. But it's a great question, and I think we can definitely do more both ways.
Thank you.
Thank you, Sharon. And John, it sounds like just capturing that as one of the challenges being communicating what the patient actually wants and us all understanding that. So I think we're getting towards the end of this whole event. Just last opportunity for those online or those of you on the panel, if there's anything you think we've missed in terms of what the challenges are to the more widespread adoption of patient-centric sampling for clinical diagnostics, now's the time to throw it in there. Just any last comments from anyone?
Yeah, I think. Yeah, go on, Rachel.
It's all right. You go first. You started.
Yeah, no, I was just going to say that, I mean, for us, patient, and I think the elephant in the room is who pays for all of this. No, it might not be as much of a problem in Europe, but in the US, where I am, and other places, the barrier for reimbursement is real. So when you are during the the pilot cases where you get it paid for, that's easy. But when it comes to implementation, it's really it's who's going to pay for the logistics, the kits, and everything. So that actually becomes a very important point for patients. But I know it's different in Europe with National Health Service. But
I think it's still universal. We've all seen examples happening within the UK National Health Service where that has been the challenge. And how do you build that? How do you build a case to get the finance? Because it's going to be, in the first place, more expensive for the lab to bring this on board. It may bring patients in, or it may make cost savings somewhere else, but to the lab, it's more complicated and it's going to cost them money. And that's one of the challenges. So I think that's a really good point. Who pays for it? Rachel, you were going to say?
I was going to say something very similar, actually. I was going to say, I think what one of the problems is there isn't the tools or the information on to make a really strong cost benefit analysis for each of these cases, because as you've just said, Neil, when you want to change the patient pathway, the cost has to come from somewhere, and obviously the cost often sits with the lab because the lab are trying to change the pathway, but the benefits are going to be across the pathway. So that for a lab itself, it can be quite an expensive thing to set up, and it's being able to confidently describe how the benefits are going to be achieved across the pathway, and then put some kind of financial some kind of figure in terms of cost as to what that benefit is. So you say, if you invest in pathology here, the wider benefit is there. I'm not explaining this very well, I don't think.
No, you're explaining really well, Rachel.
I think it's really difficult. And then when you're in the lab and you're trying to implement the test and do the validation and do all those practical things, we don't have the skills really, or we certainly don't have the experience to be doing that kind of detailed cost benefit analysis. So I just wonder how we can get round that, and I guess that's to discuss next week. But certainly for me, that's something I think would be really beneficial for whatever device you were trying to use, whatever test you were trying to implement, just to have some idea of how to sort of tell that story and give that narrative when you are requesting investment to set something up.
Yeah, no, it's a really good point. Does anyone else?
Yes, can I contribute to that point? We have to be cognizant that in many of the European laboratories that are government or within a health service, a health system, have historically had a lot of investment through the use of existing platforms. The Abbotts, the Cobasses of Roche, the BDs, etc. They are huge investments that need to be made. And so we have traditionally always said, what is the typical way in which we would use or take samples from the use of those platforms? Perhaps there's an opportunity here to think about re-engineering the amount of sample that those platforms would need to think about taking. And that touches on something that Rachel's just mentioned in the investment that is needed into the laboratories. I think it needs to be driven a little bit more further upstream in that what is the amount of sample that we will be willing to accept to use in those platforms that are already established in many of the laboratories across Europe.
I don't think it's as simple as that, unfortunately, because it's not just specifying the volume that you require, but it's the way the instruments are designed. They're designed to take primary tubes, and as soon as you don't have a primary tube, you're looking at a sample cup. And when you speak to the manufacturers, there's, in my experience, there's not a huge amount of interest, because to change one of those platforms to accept a different type of sample, I guess is a very significant ask, isn't it? So it's that, like, if we wanted to do this, if we were setting up microsampling from scratch, we wouldn't be trying to do it on a Roche analyzer or an Abbott. We'd have a dedicated platform.
Yeah.
And I guess that's why mass spec is often linked with microsampling, isn't it? Because it's much more adaptable in terms of specimen type and sample volume. And that's how I kind of got involved with it in the first place, because my lab basically does mass spec.
Yeah, let's not get too much into solution mode. We're about out of time now. So what we've got is, I've taken some notes. We'll have a look at the transcript and make sure I've not missed anything. I'll circulate those notes round the panel and the speakers as to just making sure we've not missed anything, we've summarized anything. And then we'll put that onto the LinkedIn group for people to just give their input. Are we missing anything? What are the most important things we should be looking for solutions for? Where do we get our biggest bang for our buck? What is it we should, as a group, be concentrating on? And we'll, focus on that on our April the 23rd discussion. And the idea is, coming out of that, we will be working with local, national, international groups, and within PCSIG ourselves to move forward activities. We do intend on publishing the output from this event on our website, and maybe in a publication in the Journal of Patient-Centricity, which Tim is the editor-in-chief for. But please, you'll all be receiving your invite to part 2 of this, and this is where it gets really exciting. How do we solve these challenges? What do we need to prioritize, and how do we move forwards? So with no further ado, I'd like to thank all the presenters and panelists today. I'd like to thank all the online attendees for your attention and for your wonderful questions. I apologize if we've not covered a major point that you had. We do have the transcript for this event, so if there is something we've missed and not talked about, we will add it to the notes, and that will go out in the publication, and we will consider it for what we're looking at for solutions next week. So thank you all. Look forward to seeing you all in a week's time. And for those of you that are interested in a more North American bias to this discussion, which has some different nuances, please register for the North American events on April the 30th and May the 7th. You will see some of the similar content, but some of it will be a little bit different, because obviously there are different nuances in Europe and North America. So thank you, everyone, and speak to you soon.
Thank you.